Hello and welcome. Today we'll be discussing the interesting features of the sea hare, or otherwise known as Aplysia Californica. You are going to learn why it is important and how it contributes to modern-day research. First, a little background about this simple-looking creature. You'll notice the taxonomy that the sea hare is in phylum Mollusca, class Gastropoda, order Aplysidae, family Aplysidae, genus Aplysia, and then species Aplysia californica. Its food habits are it is a herbivore, and it eats mostly algae and eelgrass. If you're looking to spot one, they can be red, brown, or green in color, can weigh up to 35 pounds, and be 16 inches long. That is one big slug. To find the sea hare in the wild, you'll have to look in specific locations in California, for it does not have a wide range of habitat. Its range is from Northern California to Baja California. Sea hares need to reproduce sexually, and they typically do so in deep waters. They are hermaphroditic, meaning that they have both male and female sex organs. If you look to the picture on the right, you'll see a lovely picture of an Aplysia Californica. Now to understand why they are important, who here thinks they have a bad memory? If you can't find your keys, remember where you put your wallet, or just can't remember what you had for breakfast, then you might have a bad memory. If this is you, then you need the memory of, drum roll please, then you need the memory of the sea hare. The sea hare, but why? This may not be the most dazzling creature in the marine environment, but it is crucial to research in memory formation. Due to the size and location of where the neurons are located in this creature, it makes them easy for collection and to see under a microscope. Learning produces long-term changes in behavior, at least in part through long-lasting changes in neural transcription. The image on the right is a drawing of what the ganglion looks like. Also in this slide are some definitions and terms that will be used during this presentation. This includes the ganglion, a structure containing a number of nerve cell bodies, typically linked by synapses, axons. The long thread-like part of a nerve cell, along with impulses, are conducted from the cell body to other cells. Synapse, a structure that permits a neuron or nerve cell to pass to an electrical or chemical signal to another neuron or to a target effector cell. Varicosities, axon endings, and sensitizations, the quality or condition of responding to certain stimuli in a sensitive manner. Now to get to the fun stuff. Scientists have been studying the morphology and physiology of Aplysia californica since the mid-1900s. When conducting scientific studies, most researchers have the objective of looking at the long-term and behavioral sensitization. Naturally, if you're going to study the sea hare, you're going to need one of your materials. Other materials differ for different studies, but they all have to be similar concepts of having handheld electric shocks. The procedure to measure the long-term behavioral sensitization is through a series of electric shock testing that is continuous for a length of time. Training consists of 4 rounds of noxious shock applied 30-minute intervals to one side of the body with a handheld electrode. Each round of shock consists of 10 pulses. The long-term sensitization was measured by the density of the synaptic connections, and the behavioral sensitization was measured by the timing of the tail-elicited siphon withdrawal reflex. The results showed that the animals had increased their sensory motor synaptic strength by increasing the amount of neural transmitters released by the sensory cells. Similar studies have been conducted throughout time, including in Bailey and Chen in 1989, to then Robertson in 2010 and Boenig et al. In 2012. But of these scientists, there is still one that is known for this procedure fairly well. Famous scientist Dr. Eric Kandel was a large contributor to researching the memory process in Aplysia, and for this he won a Nobel Prize in 2000 for physiology or medicine. Here we have a diagram of the Aplysia Californica and the training site of where they did the electroshock. So this is where the long-term sensitization of the tail-elicit siphon withdrawal reflex was. This may be a cartoon diagram, but you can see the siphon in its back. So when it would get the electric shock, the siphon would enclose inside the Aplysia Californica, and the scientists would time it from when it started to close in until it was time for it to come back out. It would only release the siphon back out when it felt the environment was safe again. Every time that it got shocked, it would increase the time in which the siphon would stay within the organism. So that's what the learning behavior was that the siphon would increasingly stay inside every time, as it would learn that the shock hurts and it doesn't want it to happen again. So it would wait until it was safer to come back out. In this slide, in the chart you can see in chart A is the duration of changes in the total number of viscosities per sensory neuron, and in chart B, it is the duration of changes in the incidence of sensory neuron activity zones. These charts compare the duration of the persistence of the memory. Sensory neuron synapses were examined at different intervals after training. These intervals are 1 to 2 days, 1 week, and then again at 3 weeks. They have found that only the duration of changes in the number of viscosities in their active zones parallels the behavioral time course for the retention of long-term sensitization. Why are we conducting this research? Because it could help humans develop medicines or procedures to combat memory loss disease, including Alzheimer's, for in the brain, the trick is to storing memories to strengthen a specific synapsis, a small gap between connecting neurons. This study has also been an enormous benefit to the field of psychotherapy and inspired researchers to take a closer look at this field. And these are my references. Thank you.